Chapter 9 discusses Mendel, heredity, and genetics. It's the first of a series of chapters that discuss genetics, heredity, and so forth. The previous chapter discussed meiosis, which is the cellular basis by which we inherit our chromosomes and our genes. Now, this review will be done in at least 2 videos, maybe 3, it depends. But we'll cover through section 4 in the first part. Now, sections 1 and 2 discuss Gregor Mendel. Gregor Mendel was a monk in the 1860s. He performed breeding experiments with garden peas. Figure 9.2d shows the traits that he looked at. Flower color, flower position, seed color, and so forth. Be sure to look in your notes and look on that. And basically he noticed there were 2 alternative forms for each trait. And he found that they all behaved in a similar way when it comes to heredity. Now, we'll use flower color as our example. Mendel came up with 2 laws or principles, the law of segregation and the law of independent assortment. Now, the law of segregation is explained best by example in section 3. Now, what he did was he developed purebred purple flower plants, purebred white flower plants. Then he crossed them. This cross is called the P generation. Okay? And when he crossed them, the offspring are called the F1 generation. All of them had purple flowers. Then he decided to cross the 2 F1s to get an F2 generation, in which case he got purple flowers and white flowers in a 3-to-1 ratio. This made him think, because whatever caused the white flower didn't go away here, but skipped a generation. Nor was there a blending effect between the 2 forms of flower color. Now, the law of segregation, again, is explained here. So he came up with several principles, and they're all outlined in section 3. Number 1, alternative forms of genes exist. We call these alternative forms alleles. In this case, an allele for purple and an allele for white. Second point, for each character, an organism has 2 alleles. See, in the previous chapter, we learned about homologous chromosomes and how our chromosomes are paired. Well, our genes are on our chromosomes, our genes are paired too. Now, he didn't know anything about chromosomes in the 1860s. Nobody did. So, an organism has 2 alleles for every feature, for every gene locus or trait. Third point, if the 2 alleles differ, one is expressed and one is not. The one that is expressed is called the dominant allele, and the one that is not is called the recessive allele, in which case they show the dominant trait instead of some blending effect or the recessive trait. And number 4, the 2 alleles separate through reproduction. That is, during meiosis, the forming of gametes, the alleles separate, in which each gamete only has 1, not 2. So we go through this example again using what's called a monohybrid cross-Punnett square. A monohybrid cross-Punnett square tracks the inheritance pattern of a single gene locus. By the way, gene locus means the location of a pair of alleles on a set of chromosomes. Okay? Or you can just call it a pair of alleles. Now, because each organism has 2, the purebred, we designate our alleles capital P for purple, lowercase p for white. It is customary to use upper and lower case for the dominant allele and the recessive allele, respectively. Now, so in the P generation, this purple flowered plant happened to be big P, and the white happened to be little p. Now, to fill out a Punnett square, first up here you separate the 2 letters from one parent. So one of the big P's goes here, one of the big P's goes here. The other parent's alleles go to the side. One goes here and one goes there. Those represent the letters that go into the gametes. The squares in the middle represent fertilization. If this gamete fertilizes that one, bring the letter down, bring it across, big P, little p. If you do this right, all 4 of these will be big P, little p. Turned out they were all big P, little p, and they were all purple, just as Mendel said. So then he crosses 2 of these. So we cross the 2 F1s, big P, little p, to get the F2 generation. Now, the rules are the same. One letter goes here, one letter goes there, one letter goes here, the other letter goes there. Recombine them in the middle. What do we have? Big P, big P, big P, little p, big P, little p, little p, little p, if done correctly. Now, genotype is the genetic description at a gene locus. We have 3 genotypes in this example. big P, big P, big P, little p, and little p, okay? One of these, two of these, and one of these. Now, when the two alleles are the same, we call this homozygous. And in this case, they're homozygous for the dominant allele, so this genotype here is called homozygous dominant. The one down here, little p, is homozygous recessive. But the one in the middle is heterozygous. Those are genotypes. But we don't see genotypes. We see traits. We see phenotypes, purple or white, in which case our phenotypic ratio is 3 to 1, but our genotypic ratio is 1 to 2 to 1 in this cross. So this is how to fill out a Punnett square and figure out the ratios. Section 4 discusses how this relates to chromosomes, homologous chromosomes. Here is a drawing of a pair of homologous chromosomes similar to that in your book, showing you if an organism is homozygous dominant, it's because one allele is on one chromosome and the other allele, which is also the same, is on the same position on the other chromosome. This is what we mean by a gene locus. Now suppose this organism is also heterozygous for the B gene, or the B gene locus, whatever that is. Well, one big B may be on this chromosome and the little b is on that chromosome. And that concludes part 1.